Tell me when you're ready. All right. Okay, so our next unit, we're going to continue our trend of sort of zooming in the cell, right? So we started kind of out, talking about what is the whole cell, right? And then we zoomed in a little bit more and said, well, let's just talk about the nucleus, okay? And how DNA moves around, mitosis and meiosis. Okay, now we're going to zoom in even further and look at the actual structure of DNA. Okay? And how does this function inside the nucleus itself? Okay, what does it look like? How is it replicating itself? Okay, and you'll see that I slimmed down some sections of this as well, particularly at the beginning. We're going to skip some of the boring history stuff. So don't be alarmed if we skip a few slides at the beginning. Okay, so this is what we are going to talk about. We certainly won't get through all of this today. But my key is we're going to do a little bit of review. Remember, we tend to start with our platform, right, our footing. So what does DNA look like? We've talked about this once before, okay, at the zoomed in level, right? What are nucleic acids? And then we're going to expand on that concept a little bit. Okay, and then we're going to talk about one individual, right, which is specifically Chargaff, right, which is an individual who figured out how base pairing works, aka A goes with T, right, G goes with C, which, as you might imagine, is one of the more important concepts that we use to understand DNA today. And we're going to find that one of our biggest roles, and I'm going to reinforce this a ton, that anything to do with DNA, like we talked about a little bit in lab, is it all has to do with opposites, right? Everything we do is going to deal with opposites, right? And that's our fallback plan. If we get lost or if we get confused, right, finding what is the opposite segment, right, is going to help us sort of recenter ourselves. Okay, then we're going to start looking at DNA replication itself, right? So what is it that we're actually doing when we look at this DNA? Now, we haven't done this in lab. This is one of the only units, although you will find a lot of the stuff we're going to talk about here is going to feel very familiar, particularly relative to transcription. Has a lot of the same features, which is nice. So there will be some familiarity here. All right, so let's take a look at our semi-review. Okay, so remember, if you're following along, we skipped all the people I don't care about at the very beginning. So we've kind of jumped probably 4 or 5 slides. Okay, so we've seen this exact slide before. Okay, and what is DNA? So remember that DNA is made up of nucleic acids, which is one of our 4 biomolecules. Boy, does biomolecules feel like a super long time ago now, right? That's what, exam 1? That's ages ago. So as we look at, and we're going to find the importance of lots of key pieces here now. So remember when we were thinking about biomolecules, we had 3 separate key pieces. My yellow and my blue here are always the same. Right? There's no variety here. Okay, so I have my 5-carbon sugar, that's my blue. Okay, remember those bends in these molecules, right, are implicitly carbons. That's an old rule we haven't talked about in literally months, I guess. Okay, well, we want to kind of uncover that if we haven't used it in a while. So there are 5 carbons. Okay, and then we have our phosphate group, right, which has a lot of oxygen. And all those oxygens make it polar and make it acidic. Okay, so if you've ever heard the phrase sugar phosphate backbone, this is what we're talking about. Okay, because it's always the same, and as we're going to see, it's literally creating sort of this ladder, the side of the ladder, right, of what we're going to do. So the only thing then that's changing or has any variety, then here's this green bit. Okay, are nitrogenous bases. So I'm showing you one example, adenine. All right, remember we have 4 of these, A, T, C, G. Okay, I'm never going to ask you to draw them out or recognize them. Okay, but understand that all 4 of them, okay, are nitrogen-containing compounds. Right, that's the key feature here of this green piece. And they all are slightly different sizes and shapes, which is what's giving us the variety here. Okay, not so bad, nice and familiar, right? Okay, so far, nothing new. We like that. It's a good place to start. Okay, so here on my right-hand side, we see that sort of ladder structure. All right, so here are my sugar phosphate. I'm drawing my sugar phosphates. Right, and you can see this would be implied on the other side. Right, so you can imagine this as like the sides or the backbone of the ladder. And then creating the rungs of the ladder. Let's use a different color here. We have our nitrogenous bases as they're paired up, right, with their appropriate base pair, right? A to T, C to G, whatever it is. So, to create our double helix, aka a structure that is bonded in 2 different locations, right? I have to be bonded here, okay, between my 2 nitrogenous bases, okay, to hold my ladder rung together. And I also need to be bonded here, okay, between the different steps of my sugar and phosphates, right, to hold the side handles together, like the different pieces of the poles or whatever really good word makes sense to call those things. Okay, so our ladder rungs, aka what's holding our base pairs together, the A to the T or the G to the C, are hydrogen bonds. And so if we look back at that previous diagram, we see there are lots of hydrogens in there. Remember that hydrogens get a little excited and bond very easily with anything that might be a little selfish and a little bit negative. We have to think all the way back now. Okay, hydrogen bonds are very strong bonds. Should have done that in pink. Fix that. Try to keep our color scheme going here. 3 times a charm. There we go. Okay. Going up and down the sides have phosphodiester bonds. We've seen these once before too. Right, fundamentally one of our covalent bonds. Okay, remember the hand holding we did with Janet and Carl? Okay, but as we see here, okay, it has a special name, remember, because it has the phosphate as the base instead of a carbon or a nitrogen, right, or any of that sort of business. We saw this with lipids. But even with that, right, we see the setup here for our old school hand holding. Ah, the days of dehydration synthesis, how ye have left us. Okay, so we're not going to go back and dig up that old business. So just knowing it's hydrogen bonds and phosphodiester bonds is fine. Okay, so doing this, right, this is how we've created our ladder, right? Rungs and side poles. Okay, so two different sets of bonds to hold together. Now, we're at a very stable biological molecule. Okay, with me so far? Okay, any questions so far about what DNA looks like? Okay, so let's talk about Chargaff then. So he's one of the few people I saw fit to continue to talk about, because what he ended up finding is one of the main pieces that the individuals that were given credit for actually putting together DNA, right, Watson and Crick, used a lot of his work. Okay, so Chargaff figured out two really important pieces of information that allowed us to start to piece together what we now know, so what you just saw. Right? And we honestly just figured this out not that long ago. Right, remember, most of this stuff is the theories of this is cumulative. Well, this cumulative piece just came, right, less than 100 years ago. It's kind of hard to imagine that. Okay. So here are the first two pieces. Now, the first piece is understanding that of all the different things in the cell, okay, DNA is the part that's varying from one species to the next. Okay, right. Otherwise, if you think about like us versus a shrub, okay, we have pretty much the same cytoplasm, the same endoplasmic reticulum, okay, the same lysosomes, right? All of that stuff is fundamentally the same. Looks the same, it acts the same, okay? None of that stuff tends to differ. But when you get inside the nucleus, okay, that material differs between us and a shrub, right? Something has to, we're different, so something somewhere in our cells has to be different. It took scientists a while to figure out what was the thing that was different, right? What's allowing shrubs to be shrubs and people to be people. Okay, so first we had to figure out what that was. Okay, so the diversity in the DNA, right? The fact that as we look at this, right, the code in us looks different than the code in the shrub, meant that DNA was probably the thing. Okay, so now it's worth looking at DNA, and so now we're investing time and money into understanding DNA. Okay, the other thing, and the thing he's probably more famous for, is as we started to break down and look at DNA, okay, what he knew is that the percentages of A's, right, adenines, were equal to the percentages of T's, thymines. Now remember, we didn't actually know what the structure of DNA was. Okay, so this was an interesting find, right, because we didn't know they were bonded together. Okay, but we knew there had to be some realistic association now, that if you had 10% A, and there was another 10% that was also your T's. Okay, so we finally figured out that these came in equal sets. We didn't know why, but it seems super important. gentleman is such a big deal, right? He's one of the 3 individuals that really laid some heavy foundation to what DNA actually looks like. Right. Hello. I haven't done these in a while. You guys look like you could use one. Whew. Kind of thought it was just going to stay gray like that for a second. Did I name it the wrong thing? Yep. Okay, so given the information that we have, okay, let's try using Chargaff's rules. Okay, so I'm telling you C, or cytosine, is 42% of the nucleotides in my DNA. Does this have like 30? Is that what we're aiming for? No, we have 25 online. So more like almost 40. Okay. 37. Which means we're about 10 of you short. So. Okay, let's start thinking through this. So if my cytosine is 42%, what else do I know? My guanine is also 42%, right? C pairs with G, right? If C is also equal to G, that was one of Chargaff's major rules. Okay, so I now know a large proportion of the nucleotides in the cell. So let's see what that adds up to. That's 84% of what's going on in the cell, right? All right, so now I have 16% left. Am I done? No, right? What do I still have to do? Yeah, exactly, right? This 16% is A plus T. Remember, we still have 2 nucleotides left. Okay, now, on the plus side, right, we know that A and T exist in equal amounts, right? So all I need to do is divide that in half. And that would be A or T, right? Both A and T, right, should be 8% each. Right, everybody see how we worked through that? Okay, so the first thing you want to do when you're tackling these types of questions is list out who's supposed to be paired with who. That'll help you and make sure that you're targeted to the right nucleotide. Okay, and then the second thing I would do is just do what we did here, right? Remember that these are supposed to be equal. Right, you can list that right out, and these are supposed to be equal. And that way you'll make sure that you've divided your numbers or percents appropriately. All right, any questions about this Menti or these types of questions? Okay, so we're going to start an in-class activity. This is going to be one you're going to be working through for a while. The top of your in-class activity, without looking back at our slide then, I want you to list both of Chargaff's rules. And remember, this can be on a piece of paper or, because I know I always say write on these, right? But this can, of course, be in that running Word document that some of you use, and that's fine too.
So someone asked when the genetics in-class activities due.
Tonight. Yep. And I updated the Dropbox for that. And some of you turned it in last night, and that's perfectly fine, right? Because you had all the information to do that. All right. Ready to take a look at this? So now what we're going to do is start looking at what does it take to do DNA replication, right? We're obviously not going to get through all of this, and that's fine. So whatever we do, we do. Okay. So the first thing I want to talk about is what do we actually need to even do this, right? What toolkit is required to start DNA replication. Okay, so as you find, we need 3 different things in our toolbox. Okay, the first thing we need is something to actually copy. Okay, and we've seen this before. Right? We've talked about needing to go to the library, right, and copy things down from books. Okay, so this is going to start our story of having things that are opposite. Okay, so when we talk about things being complementary, okay, this is a word that fundamentally means opposite. Okay, everything on one strand is the exact mirror image of the other strand. And this is where Chargaff's rules become really important, right? A is always bonded to T, C is always bonded to G. And so we always have this sort of mirror image to build off of. We know what belongs. As a result, we can use each strand as what's called a template. They're like a blueprint. Building instructions. Let's take a peek at how that actually works, okay? So this process is called semiconservative. Let's see what we mean by that. All right, so in dark blue and all the way on the left here, I have what's called our parent molecule. Okay, so just like with mitosis and meiosis, when we say parent molecule, we mean our starting molecule, right? What we've done before we started division and copying it. Okay, so you can see what we mean by complementary nature by looking at this, right? These 2 strands, strand 1 and strand 2, are always opposite to each other. Okay, A is always opposite to T, C is opposite to G, and you'll find as we get into the details of this, it even goes further than that. And so, as we go in, right, we pull apart our 2 strands, okay, and if I'm just given half of this, I only have this left half, that's left, right. Okay, I can fill that in, right? It's like a puzzle. Sudoku or something, right? I know what I need to do. So we're ignoring all this stuff right now, right? Okay, so the baby blue, or periwinkle or whatever that is, okay, are the puzzle pieces. And since everything is complementary, right, or works in opposites, I know when I have an A that I'm going to put a T in. I know when I have a C that we can put a G in. Okay, and ultimately what I can do is rebuild the strand that I pulled away. Okay, and now I've scribbled this out, but of course we do know that this happens on both sides. Okay, so we get 2 copies of the original parent, right? Daughters, which is the same thing we called with our mitosis, right? We had 2 exact copies of the original parent. So here's where the phrase semiconservative comes in. Okay, if you see here, you see I have this labeled out as 1 brand new and 1 original. Okay, the fact that we keep 1 original, right, this dark blue navy, is the conservative part. Okay, I have 1 piece that is an original. Okay, there's no way it could be messed up because we didn't do anything to it, right? It wasn't altered, it wasn't copied, it is the original piece. Okay? But then we have the brand new strand. Okay, so semi. Okay, so this part is brand new. Now, we'll eventually talk about all these different checks and balances, so it's unlikely this is messed up. Okay? But if there is a mistake, it's going to be on this side, right? Because this is the side we hand built. Okay, make sense? All right, what else do we need? So the other two things we need work in cahoots together. Okay, we need a bunch of enzymes that are going to work together. Okay. Enzymes that are actually going to do the work. I like to think of these like construction workers on the job. Okay, primase, as we'll talk about, right? Think like primer, like the first kind of paint you put down, right? Primer on a lawnmower. I don't know that those really have that anymore. Okay, but primer means to start. Need you primed. Okay, so primase is the first enzyme, right? The first construction worker that's going to come through. The second one that's going to come through, and we'll find there's actually 3 different ones of these, it is DNA polymerase. Okay, so you see both of these have that A as the ending, right? They're telling us they're enzymes. Okay, this does most of the work. Okay, so the construction workers that stay there and do the bulk of the job, the digging, the laying of the tar. Okay. The other thing we have, okay, are the tools, right, or the items we need to actually do the work. Okay, so in this case, shovels, cones, tar, right, all of that sort of stuff. Okay, and in this case, we need both RNA, as you'll find, right? We're going to lay those down as a start, kind of like a little runway. And then otherwise, largely, just DNA nucleotides. And that's going to be most of the gear that we need. Whatever, I don't know that I care today. Okay, so you are not going to turn in your DNA replication in class. Hang on to that till after the test. And we're going to continue to add on to that as we start to build our own DNA strand, which is a little bit longer than usual, so make sure you tuck that or save that right in a very safe place. Okay, as always, please make sure you're doing your Socrative. And then don't forget, this is a big, busy weekend, right? It'll leave you with very little to do for biology for a couple weeks, which is kind of nice, right? We like that. There's a lot for me to do, but very little for you to do. But we don't want to lose track of either our paper, right, or the test. And don't forget, there's 2 different review games running right now, right? The genetic Socrative, okay, and the quizzes. So use all your resources if that's your stick.